So hi everyone, thank you very much for joining us today. This is the Cancer Center's Resources for Research seminar series. We're very happy to welcome two speakers today from the Columbia University Biobank. And to introduce the speakers, Dr. Murdoch Riley will tell us a little bit about our two speakers. So Dr. Riley.
Good afternoon. It's a great pleasure to actually be invited to introduce Wendy Chung and Christoph Kirchhoff to present on the Biobank today. They will provide some of the context for this resource. But I just want to point out first that this is a resource that's generated for the use of the community. It's a nascent resource. We're kind of getting it up and running, so there's still challenges in doing so, but there's a lot of progress being made you'll hear about. And then Wendy and Christoph have been amazing leaders and drivers of the work that's been done so far, and really have dedicated themselves over the last 6 to 7 weeks and even longer towards this vision and this mission. So this is something that falls under the school and the Office of Research for the benefit of campus. Is a collaboration between the Office of Research and multiple entities, including the Irving Institute, IGM, multiple departments, DBMI, the Cancer Center, and others. So as you know, that Wendy is a star human geneticist and clinical geneticist on campus, and Christoph also in kidney disease, both on Mendelian disorders and complex disorders in kidney and complex traits. They both are leaders of the precision medicine resource in the Irving Institute, but independent, and the CTSA, but independent of that, they're national and international leaders on human genetics and precision medicine. I'll let it go at that. There's a lot more that could be said, and let them get started.
Great, thanks, Murdoch. So Christoph, you can go ahead and advance the slides. So what we're going to talk about today is a...
So Wendy, Christoph, you need to unmute yourselves and
Very good. So we're going to talk about today is this precision medicine resource that's part of the CTSA across the university, specifically now focused on the COVID portion of this. I think there are a few guiding principles that I wanted to instill. We've tried to do this, and we got this up and running very rapidly with a large number of people, and we'll give credit towards the end, but just a large number of people putting a lot of energy into this. We started out with a few sort of guiding principles in this, which is that this descended upon us very quickly. There were issues in terms of being able to develop this and keep everyone safe. That is, that patient care was always first and foremost, and we didn't want to interfere with that. We didn't want to use extra PPE in terms of our recruitment staff, and dealing with these specimens requires specific biosafety. And so in doing that, we were trying to do a lot of things for a lot of people, but realizing that this is difficult under the circumstances we were doing it. So it's not perfect, but we'll share with you what we have. Again, we're going to focus today on the COVID-19 positive patients. This is defined by, as best as we can, those that were PCR positive at the time, where we've been able to identify them through the EHR. And remember that we just went into EPIC 5 weeks into this, so it's not perfect, but we tried to identify individuals who are diagnosed COVID positive, be they diagnosed at other laboratories or transferred in, and be able to include all of them within this. As we did this, we took discarded biological specimens, so individuals who had clinical samples for instance, a CBC or nasopharyngeal swabs, and we tried to use the principles of recycling this precious resource to be able to identify for those individuals, put them aside and use them either de-identified or consented for the community. There are some limitations that come from this because clinical samples need to be used for the clinical tests. They need to be on standby, usually for 3 or more days, in terms of making sure that if someone needs to go back and use that specimen again, that it's available. And so these are not pristine the way we use them for regular research. So just appreciating those limitations. We did try and be very broad, though, in terms of the types of specimens that can be used, and we'll give you some numbers in just a second, so that, again, as much research could be done with these samples as possible. There are some requirements in terms of how we had to treat the specimen to inactivate the virus, and so we tried to make decisions about making it useful for the most people in the most ways, but again, it's not perfect. As we're thinking about who's to use these samples, one of the guiding principles is obviously we want Columbia researchers to be able to use them. We want data that we generate from this to be useful for many people, so hopefully data that are generated are useful across multiple investigators, and also to collaborate beyond Columbia, because we realize that many of these questions for sufficient power are going to require more than a single institution, and just to make sure that everyone gets credit where credit is due. Next slide. So as we're doing this, this is part also of a larger biobank mission. So we had been thinking about this well before COVID descended upon us, and that was good because we had a lot of the infrastructure in place for the Columbia University Biobank. This is something that, again, a lot of people have put a lot of investment and resources into. It's been run through the Irving Institute and the CTSA, and in part, set up so that we could piggyback and not interfere with what many of you know as being the NIH All of Us research program. That portion of it hasn't actually ramped up yet, but once we get through the COVID era, we will get back to doing some other research, including All of Us, and piggyback or jointly recruit for both of those studies. Next slide. So with this, there are a lot of people who have contributed, and just to be very clear, we've had discussions first and foremost with our patients in the community to understand what they wanted from us and understand how they felt about this. And so we've had actually even a discussion. We had 80 people online with our community in Harlem last week in terms of getting feedback about all of this. And so it's been great to hear feedback from the community with this. Many different groups within clinical pathology. I just want to put a shout out specifically to Eldad Hod and the system that he has set up to be able to use and reuse these samples, biomedical informatics, and then across the clinical areas, really almost every clinical service has had an input in thinking about this, both what the needs are and clinical curation of data. It's been like nothing I've ever seen in my 20 plus years at Columbia. So just credit to everyone for what they've done. And it's definitely been a team effort. Next slide. So in terms of doing this, it's complicated. I won't show you how all of the sausage is made exactly, but we are taking the data in terms of knowing who's COVID positive. We're doing this about 3 times a week to identify who those individuals are. Those specimens, as I said, within pathology are being set aside. Simultaneously, we have a core of about 20 individuals who are managing going back to participants, to patients, to be able to get consent from them. Largely, we have been focusing on patients once they're discharged from the hospital. Again, we made this decision early on in terms of what was putting minimal burden on, as you can imagine, very anxious patients and families, very sort of complex situations within the hospital. And so the vast majority, although not quite everyone, is consented after they're discharged home for the hospital. As I'll show you in a second, unfortunately, not everyone has survived this condition, and so we do have a fair number of fatalities as well. With that, the autopsy service, and I just, again, shout out to Jim Goldman, who's been managing this. The autopsy service has also been taking banks of tissues. Again, there have been variable periods of times after individuals are deceased, but there is an opportunity for those of you who need specific tissue samples to be able to use those. And again, I'll get to it in a little bit, but how to apply for this information. With this, the types of samples are included, as I said, things that would be seen in the normal clinical pathology workflow. So things like CBC, serum, plasma, urine, even a small number of samples, but in terms of fecal material, small number of CSF samples, but largely these are blood samples. We've also, to ensure that we could do this in a single streamlined flow process, been able to extract DNA, genomic DNA, from a large number of specimens for genetic studies, and centrally those data will be generated and shareable to all of the investigators who would do that or use that as part of the analyses that they have done. Individuals who are consenting to this study are also consenting to the larger biobank. So although this information is being used first and foremost right now in terms of COVID research, they also are available for recontact. So other studies that we might, or you might invite them to, and thinking about, for instance, outcome studies, either outcomes of COVID or other things that are related to health generally, but they are part of now our larger Columbia Biobank and available in terms of other studies. The clinical data that we have is largely being driven by DB&I in terms of the data polls, and we'll get to it in a second in terms of how you can request that data, but there's a data mart in terms of the data that are coming in, and also a shout out to a huge core of people that are out there hand curating data through some of the clinical working groups, really doing very fine curation to be able to make sure that we get the information right clinically. Next slide. With this, we wish we could go faster for everything that we're doing, but as we're doing this, this is set up as having a steering committee and then a group of working groups that are thinking about individual aspects of this, a genomics working group, a patient enrollment group, things dealing with operations and management, but individuals thinking of all of these pieces. And Christoph is going to talk specifically, but he's been working really hard in terms of the samples themselves, getting the samples, inventorying the samples, and making sure that they're processed and stored correctly in the bank. Next slide. Within this, to be able to use the samples, we've tried to make this process as easy for you as investigators as possible. We'll have a website that'll be a little bit later on, but the initial application is very, very simple. It's literally a one-page application in terms of doing this. Depending on what you need, you may need IRB approval. You may also need to think about, if you're using the specimens, specific issues in terms of just safely using those specimens. So environmental health and safety will be working with you closely to make sure that you're protecting your staff and that you've got the right environment to do this in. And then we've been, in fact, doing pre-reviews and then committee reviews for the request to make sure that as much as we can make sure that people are getting what they need in terms of both data as well as specimens. And in some cases, helping investigators think more carefully about the quality of the samples we have and whether or not adjustments need to be made to their plans, because we may not have things that are sufficient or ideal for what they're planning to do, and or they may find that there may be other individuals with whom they can collaborate, so that if, for instance, they're both interested in the same interleukin or something else, that there may be some economies of scale with collaborations and studies that are going forward. As I said, there are specific groups that are thinking about particular groups. So there are, just to give an example, individuals in OBGYN that are thinking about the women who were positive during their pregnancies and then following the outcomes of those pregnancies. And so that's just one particular use case. You can think about within the Cancer Center, for instance, identifying anyone with a prior diagnosis of cancer and looking at that subset of individuals and being able to see what their outcomes were, people who may have been on chemotherapy or other things to suppress the immune system. So we can certainly answer questions in the Q&A, but there are ways that you might be able to use the samples, and you may not need the entirety of the sample, but some subset. We do have a commitment, and again, this goes back to our community engagement. We do have a commitment and had the option for individuals who are participating within this to be able to get information back. So the plan is to be able to get aggregate information from what we're learning as a community to communicate this back to our participants as a whole. But even there's the option, if we learn something, for instance, genetically, that is something that is what I call news you can use, but something that is medically actionable that would be helpful to them, that they can get that information back. And again, thinking about the Cancer Center specifically, this could be things like cancer predispositions, like hereditary breast ovarian cancer or Lynch syndrome or things like that, that if we do, although it's not at all the primary focus of this COVID research, when we do generate data, if those types of things are recognized, participants do have the option to get that information back. Next slide. And Christoph, just remind me, I forget at what point we hand off, but just jump in at the right time. I think maybe this is my last slide, but for the working groups, again, we have a lot of experts, especially clinically, thinking about all the different areas that are affected. And I think many people know, but cardiopulmonary, renal, are sort of hardest hit with much of this, but we also do see manifestations with many other things, a little bit with strokes and neurology, a little bit in terms of rashes. So dermatology even has some role, and then again, age-specific groups that are coming in, and of course, all of this being related to infectious disease. Next slide. Should I do this one, Christoph? I forgot, I'm sorry. And you're on mute.
Please go ahead, yes.
Okay, so just to give you a timeline of when all of this started, it feels like, I swear to you, it was 2 years ago, but it really was not that long ago. When March 20 is when we started, we officially had IRB approval to march through this. And since then, we've been essentially going backwards and getting the samples that we had, getting the consents that we had for patients that were in the hospital prior to that time. As of April 1st was the time we had first people officially enrolled and consented. And since we've done that, again, just a lot of credit to a lot of people working literally 7 days a week on the project, trying to play catch up and keep up with everything that's been going on. We have most recently, when we started to appreciate that some of the samples that we had that were discarded samples, perhaps not, well, in fact, we know not being ideal for some of the things that investigators wanted, we have been trying to adapt the protocol to meet your needs, and we continue to want to hear feedback in terms of what you need. Starting mid-April, we realized that we would need to consent patients who were still in the hospital to be able to get, if you will, fresh samples that would be useful in terms of, for instance, RNA sequencing or other types of specimens. So we do have a relatively small number of inpatients that we are consenting while they're inpatient to be able to allow for that part of the protocol. It's just appreciate it's not easy for the individuals and their families, so we're not going to be able to do this for everyone who's inpatient, but we are trying to meet the needs of the investigators for those sample types. Next slide.
Okay, so I think I can take over now. So this is a collapsed version of kind of major components of the biobank. There's a clinical component to this that involves interaction with the patients, and we worked really hard to work out a safe way to do so, implementing electronic consent mechanism, and also we designed all of our data collection tools for self-reported symptoms and clinical data in the form of electronic questionnaires. The second part is the biobank, and this part is divided into a liquid biobank, which consists of discarded specimens, as Wendy has described, and also more recently, a tissue biobank that includes autopsy tissue. The third element is the COVID-19 data mart. This is a major effort from our Department of Biomedical Informatics that collects and curates all of the electronic health record data from patients who have been diagnosed with COVID at our institution, and also merging this data with self-reported questionnaire data provided by the patients. And finally, there is a genetics and genomics component where we are actually trying to generate data from the collected biospecimens. So obviously, when we started this, both myself and Wendy were very much interested in genetic questions related to COVID. And one of the really motivating factors was the observation that there's really a very striking inter-individual variability in the disease course and also in the complications of the disease. This relates to the severity and duration of the disease itself, including symptoms of cytokine storm, and the fact that some individuals are completely asymptomatic and go through this infection with minimal symptoms. Of course, for some of the most serious complications, such as pneumonia, respiratory failure, death, also acute kidney injury, are the ones that we are worried the most. And here, again, we're seeing a lot of inter-individual variability, and we suspect that perhaps there are genetic factors that underlie some of this susceptibility. There's also predisposition to cardiovascular events, MI, strokes, arrhythmias. There are a lot of arrests happening in the hospital among patients who are hospitalized with COVID. A lot of thrombotic events have been described, and also some rarer complications, including myocarditis, pericarditis, hepatitis, gastroenteritis, some autoimmune phenomena, You might have seen the article in the New York Times today about Kawasaki disease and pediatric patients affected with COVID. Again, very rare complication of the infection, but clearly there seems to be a pattern of predisposition among certain individuals to develop these type of conditions. And other conditions such as anosmia, agwesia, and also neurological complications, including encephalitis. Some of the earlier observations were also that some of these severe complications, including respiratory failure and death, were aggregated in families. And that type of clustering in families made us think that perhaps there's a strong genetic predisposition to some of these complications. And finally, there's also clearly there are differences in susceptibility by race and ethnicity. And this is a little bit difficult for us to figure out from a genetic standpoint, since obviously there are also economic and other disparity factors that might be in effect here, producing some of these differences. But of course, the question of ancestral differences in susceptibility is also on our mind, and we should be able to address this question by combining socioeconomic data with genetic data. Clearly, a lot of these analyses, a lot of these phenotypes have age-dependent expression, and there's also well-known sexual dimorphism where males are more predisposed to these conditions or some of these conditions than females. So these are factors that we'll have to control for in our future genetic analyses. Of course, when we think about performing genetic studies on these cohorts of patients infected with COVID, we really need to harmonize our phenotypes. And for this purpose, early on in the effort, we designed a self-reported data questionnaire that consists of 76 questions related to symptoms, things like socioeconomic status, things like even anxiety scores, and basic information about treatment, medication exposures, and risk factors. This questionnaire has been now translated actually into 9 different languages, actually 8 languages in addition to English, and disseminated across other consortia who are doing similar type of research. The way that we implement this questionnaire is actually in the REDCap. Again, it's an electronic delivery to the patient, and the patient completes the questionnaire online. And we also shared the code book and data dictionary for this questionnaire online, and you can see a GitHub link if you're interested in finding out more information about this. In parallel, the second clinical data stream for this study comes from electronic health record data, and I already described, and I think Wendy has covered this as well, the major effort coming from the Department of Biomedical Informatics on the construction of COVID-19 data mart, which really synthesizes a lot of the information that is automatically derived from electronic health record, but also hand curated by a number of medical students who are involved in this effort. And ultimately, we are planning to form phenotype groups that will address specific phenotypes in genetic studies across many different subspecialties of medicine, and also including pediatrics and obstetrics gynecology. So let me tell you a little bit about the liquid biorepository. So as Wendy mentioned, the liquid biorepository includes, basically involves collection of discarded biospecimens. These are specimens that are collected for routine clinical testing. So for example, if a COVID-19 positive patient is admitted to an inpatient service and gets a daily CBC, a leftover EDTA test tube is being saved for the purpose of the biobank. There is a bit of a delay between the availability of that test tube for the biobank because the clinical lab is holding on to these biospecimens in case they need to rerun the tests or if there's some add-ons to the original order. So these specimens are perhaps suboptimal for certain types of studies. For example, if you want to rescue live cells from buffy coats, these specimens are really not ideal for that. Similarly, because of the delay and the fact that these test tubes actually sit on room air in a clinical lab before being processed by our biobank, they're probably not ideal for recovery of host RNA, for example, from immune cells in a buffy coat. We implemented an honest broker system where these de-identified samples are paired with electronic health record data by an independent entity, and that function is being served by the biomedical resource of the CTSA. And finally, as Wendy has described already, we are performing remote e-consenting for genetics, and we have 20 coordinators across many different labs that contribute to this effort. Of course, a lot of clinical studies are on hold right now, so we are benefiting from kind of free time of many coordinators who donate their time to recruitment for this particular study. And then, of course, we collect self-reported data, as I described before. This is just a snapshot of some of the numbers that I have. Actually, I don't have the numbers from this week, but this is as of 4/26. You can see that we have multiple samples. In the first column, you see number of actual samples that are stored. And then on the right-hand side, you can see these samples mapping to unique patients. At this point, we have probably over 2,000 buffy coats from unique patients. We also have a fair number of urine samples. Some of these are repeat samples. And we also have a large number of nasopharyngeal swabs. This number is increasing rapidly because the testing with nasopharyngeal swabs continues. And so the actual number this week is actually much higher. And we also collect plasma and serum. And you can see the numbers here. The plasma and serum pretty much, in terms of number of samples, overlaps with buffy coats. There's slightly more serum samples. And again, there is a large degree of overlap in these biospecimens. So for some individuals, for I would say many individuals, we have multiple biospecimen types, which is really great if you plan multi-omic approaches. And so for example, for genetics, we're very much interested in overlaying genetic data with, let's say, biomarker data from plasma or serum. And if we can also generate transcriptomic data, for example, for some of these samples, for some of these individuals, the data set becomes so much more powerful. We have established an efficient DNA workflow where we take these buffy coats and we basically transfer them to a DNA extraction lab. And you can see as of today, we have extracted over 2,500 samples. And generally with DNA, we're getting very good quantity and quality of DNA. So these samples are actually ready for sequencing. So what's our plan in terms of data generation from this biobank? As I mentioned, we were initially very interested in host genetic data. And so certainly here we want to cover both the rare variants and common variants for different types of analyses. So all of these samples will undergo whole exome sequencing. And also we agreed to go ahead with low-pass whole genome sequencing, which is a newer method to generate SNP data, and perhaps it's a little bit more accurate compared to the SNP arrays, and it perhaps is a little bit cheaper. So this is our first line approach for generation of genetic data. We are piloting generation of host transcriptome data from the leftover nasopharyngeal swabs. But the problem here is that a lot of these swabs coming from the clinical lab actually sit on room air for a little bit too long. So we're not sure if this is going to work out. At the same time, we can probably recover viral genomes from the nasopharyngeal swabs. And again, here we are running pilots to assess the feasibility of this. And of course, if we have both the viral genome and the host genome, then we could think about really interesting studies assessing viral host genome interactions. So this again becomes much more interesting from the type of scientific questions that we can ask. Also, because we have nasopharyngeal swabs, we are preserving DNA for nasopharyngeal microbiome studies. So in terms of study designs for host genetic questions, again, we're gonna look at, the plan is to generate exome sequence data, look at rare variants. Specific phenotypes of interest here would be early onset disease, looking at kind of the phenotypic extreme of pediatric patients with really severe disease, so unusual cases, looking really for kind of monogenic forms of defects in immune system that trigger more severe presentation of the disease. We have a plan to extend our recruitment to families, especially those with multiple affected individuals, to look for familial mutations that might be segregating in these families, co-segregating with more severe complications. I should say the exome sequencing is being done in collaboration with David Goldstein at IGM. And of course, he has a pipeline for gene-based collapsing association analyses. And here we're planning a variety of different approaches, looking at case-on analyses, assessing susceptibility to specific complications, but also kind of larger scale population-based approaches, where we just look at the susceptibility to the viral infection. So comparing the sequence of affected with population controls, and that's a kind of design that's also being taken by some of the other consortia that are looking at the very similar questions. In the common variant space, we're interested in GWAS for complications and a case-on analysis, and of course susceptibility, which is also a population-based type of approach. We're interested in trying to disentangle the ancestral effects and doing admixture analysis on these samples, and analyzing the genetic ancestry in combination with socioeconomic variables, trying to see if there is specific, genetic effect coming from ancestral background. And I also mentioned host viral genome interactions. There's great interest in HLA region and HLA effects and susceptibility to the virus. And if we can recover nasopharyngeal transcriptomes from the hosts, we might be able to also look at nasopharyngeal EQTLs, which might enhance the interpretation of some of our data. And of course, we need to account for age and sex effects, looking for age and sex interactions. And anything that we do in this space we realize will really need replication external cohorts, since even given the fact that we actually have probably the largest number of samples in New York City, still for genetic discovery, the sample size, especially for some of the rare complications, is relatively small. So it will take a bigger collaborative effort with other national cohorts and perhaps international cohorts to get these studies done. And so here we're planning to collaborate with the New York Genome Center. We have formed a national CTSA network of all of the CTSAs that have a precision medicine resource and that are doing similar things. And we are sharing our experiences and sharing protocols. We are sharing the questionnaire tools in order to accelerate our biobanking efforts. And then the plan is for us to combine forces at some point and cross-replicate our findings as well. We're also part of the International Common Disease Alliance Network, which formed a global effort targeting host genetics. And we're hoping that our data will also contribute to this larger international effort. So I think with this, I'll pass it back to Wendy to talk a little bit more about the recruitment and consenting process.
So as we've been doing this, we have bilingual coordinators who are able to consent in English and in Spanish. And at this point, we have, as of last night, 593 who had provided full written consent. There are another approximately 400 who have verbally consented, but we are still in the process of getting the written consent from them. And unfortunately, we're close to 700 individuals who are deceased. Next slide. As Christoph said, we had implemented a questionnaire. Next slide. This is all online. It's in both English and Spanish. And the questionnaire completion is running right now a little bit higher than this, actually about 86% complete. That actually has detailed information, as I said, that's beyond what's in the medical record. We're happy, as Christoph had said, the survey is up on GitHub. It's in many different languages. And so whether you're using the data from our study or you want to apply it to a different cohort, we specifically wanted to make sure it could be very inclusive and many different communities could use this. Next slide. In terms of now how you actually get the data, and this can be either clinical data or if you'd like to be able to get any of the biospecimens. For those of you who have previously put in track requests, you're familiar with this in terms of being able to get clinical data. You can go simply to put in a track request. I will warn you that there are many people in line to be able to get clinical data, and so you won't get your track request fulfilled immediately, but that's the shortest and fastest way to be able to get clinical data only. For an application for biospecimens, any of the things that Christoph or I have talked about today, the website is here. It's a one-page application, and if you have specific questions as you're thinking about whether or not something is feasible, whether or not the specimens are going to be good enough, you're certainly welcome to send an email to the centralized website. You can ask us questions. It'll just be a couple more minutes and you can ask us questions here, or you can certainly reach out to any one of us. I will say that although the number of samples that you may have seen is large, to appreciate that's for the whole community to use. And so also thinking about realistically, what you can do in terms of minimal number of samples will help to make sure that the most people can use the samples as possible. Next slide. So this just goes into a little bit more detail in terms of a couple points that I wanted to make. I think many people didn't think about it initially, but many of the biospecimens we're talking about, at least at one point, have had live virus. We have inactivated many of the samples so that they will be safe, but depending on what you want to use and how you want to use them, the university is in a good way trying to make sure every one of the workers in our laboratories is going to be safe. So as you're going through your IRB, there's Appendix A's that you'll need to worry about in terms of environmental health and safety. These are considered in terms of handling BSL-2, and so they're just both training for your staff as well as particular infrastructure you'll need in your laboratories. And again, you can go to EH&S and there's information on the website as well. Again, if you need prospective recruitment, please feel free to reach out to us. We're doing what we can for individual investigators, especially if the number of samples is not too large. And then Jim Goldman is handling the autopsy cases in terms of tissues that you might need. Next slide. With this, depending on your study, you may need IRB approval. If it is possible for you to use de-identified samples, again, we can provide both de-identified samples and de-identified data. So think about what you need and whether what you'll need in terms of IRB approval for that. As I said, we're trying to do as many things as we can for those of you that are putting in grants based on this, administrative supplements, anything you might need, be able to provide letters of support, at least to get things going. If you do need preliminary data on small numbers of samples to show feasibility, we are trying to prioritize those, and again, trying to make sure that the best science is done, and that if there is something that will allow for data sharing for other investigators to use some of the same data that you're generating, make sure we don't reinvent the wheel as we're trying to provide these resources. Next slide. So in terms of how long things take, letters of support for your grants, we should be able to get back to you very quickly. So we have template letters for those. So if you need those are relatively quick. We have met in terms of being able to grant data or sample requests. So some of the easier sample requests are going to be going out soon. This process is just starting though, so these samples won't come to you overnight in terms of when you'll get your samples. And again, some of the samples, if they require prospective recruitment, you will need to speak with us to be able to figure out the study design or individual clinicians with whom you might collaborate. And I think we just have maybe one more slide of acknowledgements. So within this, and this is very incomplete, but just to say it's taken a huge team of people to be able to do what we've done to date. And we just couldn't thank everyone enough. Everyone has really volunteered, and this hasn't been our day job, but we've wanted to make sure that this could be done in an organized way to help as many people as possible, and of course, ultimately to help our patients. So we'll be able to stop here, and there may be some questions on the chat or other places that we can start looking at.
Thanks so much, Wendy and Christophe. Just before we get into questions, please everyone check your chat box, and if you have a request to identify yourself by uni or email, please respond. Some of the names on the screen are ambiguous, and we like to follow up with everyone who's attended. We do have some questions. Here's one. If we have a list of patient MRN, IRB approved, that we would like to get clinical data known to be available through DataMart, does that go through track requests?
Yes.
Yes, absolutely. So this would be a very simple scenario where you just apply through track and get access directly to the data.
Great, thank you. Is the autopsy bank form available in the Biobank website?
So the form is there, that same one-page form is what you use, and then Jim Goldman is the one who receives and reviews those applications, and we'll get back to individual investigators.
Great, okay. Another question, core data that is being planned to be generated like RNA sequencing, would that be what would be the timeline when we can expect it to be available?
So maybe I can answer some of this this particular question. So for genetics, the DNA is being extracted right now, and actually this week we're starting exome sequencing. The plan is to deposit to basically have the data available in a central resource for download within 2 weeks of data generation. So pretty fast timeline. For RNA sequencing, we're still in the phase of piloting different approaches, and it seems like the best approach would be to collect fresh samples prospectively. So this obviously will take much longer to accomplish. Thank
you. Are there any other questions? Please feel free to unmute yourself or pop your question into the chat box. I don't have any other questions here. Here's another question. Can you please post how to submit a track request?
So I see Ben is online. Ben, if you happen to be able to, would you mind putting that up? And you just did it. Thank you.
Great, that's up. That's perfect. Does anybody else have a question that they'd like to ask? Now is your chance. Please feel free to unmute yourself or pop your question into the chat. We'll give it a minute. Nope. Okay, well, I'm sure that, go ahead.
Let me just say, this is Wendy, let me just say very briefly that we are trying to think about what the community is going to need as this goes on. And I will say that, as you can imagine, we're skewed towards those who've had the most severe outcomes. So those who have passed away, those who've been admitted. And if you think about who actually got admitted in March and April, it was people who were pretty sick. We're continuing to expand now that serology testing is going on. We will identify individuals who were COVID positive but recovered. And so those individuals will also be available to be recruited. At some point, we will have to think about resources that we continue to put into this and how important it is to the community. So we are interested in hearing back from you about things that either would round out the cohort and gaps that you feel would be high value in terms of filling in gaps. be they participants, be they samples, be they data that we gather, outcome data, follow-up data. So we would love to hear from you in terms of what your particular outcome interest might be. It'll help guide us in terms of making decisions that are good for the community.
Thanks, Wendy. We have another question here. Once you have club approval with the appropriate IRB and Appendix A, who do we contact to obtain SERA? Is it LDAD, HUD, or somebody else?
So you should.
Yeah. So this is yeah. Go ahead, Wendy.
I was going to say, so we are trying to do this centrally. If you put in that brief one-page application, then we can make sure that that gets distributed equitably to the community. Those are the same samples that were coming from LDAD, so you don't have to bug him so much. It relieves some of the burden off of him.
Yeah, I think the only thing that I want to mention about Serum is this is one specimen that LDAT is still holding on to because of the need for validation of different serological tests that he's doing in the clinical lab. So we haven't really addressed the distribution of Serum, but we are addressing right now the distribution of plasma at this point. So, you know, if you absolutely need Serum as opposed to plasma, you know, I think there will be some delay in terms of our ability to provide the specimen.
Thank you. Is there any plan to collect saliva as well?
So I'd be interested in what the community might use the saliva for. We do realize that there's certain individuals who've been consented, but we don't have either sufficient or high enough quality DNA, and we would go back to send kits to their home to send us saliva samples back so that they could be part of the genomic and genetic studies. There are also individuals I know that are thinking about using saliva as a way of validating or doing serologies from home. They require slightly different specimen processing and specimen types. So the more detail you can tell us about why you might want saliva, is it for microbiome, is it for DNA, is it for ELISA testing? We'd love to hear what your specimen requirements are, both how many and what type and for what purpose.
Laura, do you want to?
I just want.
Go ahead.
Yeah, I just wanted to add that, you know, saliva is not being used as part of routine clinical testing, so it's not available as part of our biobank right now. So if you need saliva, this would involve some sort of prospective recruitment, and then we're open to partnering with you if you're interested in getting that particular specimen. But we'll have to really find out a way to do that.
To the question answer, Flora, do you want to unmute and discuss, or did that answer your question?
Hello, thank you so much. Yeah, I think there is like a kind of debate about using salivary diagnostic to be able to look at the viral load or antibody, and this is something that can be useful if you have like saliva sample collected as well.
So as Christoph said, we don't have saliva samples right now in terms of doing diagnostics with acute infection or being able to take the virus. I do know of people working on saliva as a method for instance, at-home serological testing. But as Christoph said, we have plans for doing some things from home with saliva samples, but we haven't yet started or implemented any of that. So again, Flora, if you want to contact us with, you know, if you're interested specifically how many you would need, if you'd need paired serum samples to be able to test the serologies for validation, or what you might need, certainly reach out and let us know.
Thank you so much. I will send it.
Okay, we have another question. Will whole blood or RBC dense blood infiltrates after serum plasma separation be available for RBC membrane analysis?
Yes, so that's not something that we've been collecting, but that's a good question. We would be interested in talking to you so we can perhaps preserve some of the red blood cells for your type of analysis. But we're not preserving it right now.
All right, we hadn't thought about this use case, but it could be done.
Okay. Thank you. So are there any other questions from the group? This is, okay, I think we have another question here. Are you unmuted?
Yeah, this is Murdoch here. I'll just thank Wendy and Christoph and add to what they've said that, you know, we will probably have some limited opportunities for prospective activities And when we would do such a prospective activity, trying to capture requests and needs would be part of that. So we would like to hear people put informal requests into the biobank. When you look at the biobank online site, there's a formal request form and there's an informal request form. So if you've got ideas and you want to float them by, then send them to us. This doesn't mean we can do everything. It means that if we're going to do some prospective work, we would be able to try to maximize the approach for that. So gathering information on that would be great.
Okay, we have one more question. Is there placental or fetal membranes from OBGYN?
So within the CUB Biobank, we haven't been collecting them, but if you're interested within OBGYN, I think many people know that there were some complications with individuals who are COVID positive at the time of delivery. So there are investigators in OBGYN who have done this, not necessarily for every delivery, but there are specimens. And if you talk with the research unit, I know you can speak with Ron Wapner and he can put you in touch with the right people that have been working on this.
Yeah, I think one comment on that is that Ron and Anna Penn from PEDS have maternal or mother offspring pairings and materials, different amounts of materials collected, and part of that is with pathology, and actually part of it is in a REDCap database linked with us, but it's not formally integrated yet. So we're meeting this week to try and have conversations about how best to integrate and how to do that. So it'll either be through Ron and Anna or through the biobank that some of those resources are available. We don't know what's in that yet, really. We don't really know what's fully in that and what the different types of material.
A couple more questions following on from the placental and fetal membrane question. What about other tissues? And someone else asking about fecal samples.
So I can answer the fecal samples. They are actually being collected, but only fecal samples that are being sent as part of routine clinical care to the clinical path lab for testing. So for those, we do collect them. I can't give you the exact number right now, but it should be probably somewhere around 100 samples right now. So it's not a large collection, but we are holding on to these. In terms of other tissues, that's really the autopsy effort that's being led kind of separately from the liquid biobank effort. But same application through the CUB Biobank website would capture these tissues. So you could, again, the number of actual autopsies performed is fairly small. So we're not really capturing other tissues to the same extent as we are capturing DNA and blood samples and urine and so forth.
Yeah, I'll add to that just to say that this is one area where the community came together really well, particularly pathology, but Emily Isai, who's on, I think, can speak to this. James Goldman is the chair of the tissue and autopsy committee, and they're performing rapid autopsies as in the current time to satisfy research requests. There's a formal process. There's a committee that Emily's on. Emily led the way really in defining some of the best practices working with other institutions. So there was a tremendous group effort to put that in place. So there are tissues that are collected. You can put in a request and they'll be considered for autopsies that would be rapid autopsy or fast or warm autopsies. I think also then on the thesis there's 140 is what the last I heard. Not sure if that's individuals or total number of samples, but it's a modest number.
And then for the question of CSF, there are a very small number of CSF samples that have been collected. My recollection is it's about 14-ish, but it's not very many.
Okay. And a question here about CSF, is that being collected?
Just a few CSF samples.
Okay, great. And I'm sorry, you answered that. And Gulam Manji asking, any sequential specimens being collected from prospective outpatients?
There is a number of sequential biospecimens, but it's a relatively small number, and it really kind of depends on the biospecimen. So, you would have to give me a little bit more information in which biospecimen you're interested in. Of course, for DNA recovery, we just target, recover from, one specimen per individual or two for backup, but not sequentially. So it really depends on the specimen.
And again, Ghulam, cancer patients on treatment, nasal, he's answering that nasal swab serum. Those would be the samples. Ghulam, do you want to unmute yourself and discuss?
Yeah, hi, thank you for really organizing this.
Sure.
We have a prospective study that is currently being reviewed by the RRB, where we are having a difficult time, trying to understand how long the cancer patients keep the virus, and when would it be safe for restarting them on treatment, and there's really no data available. So it would be really helpful to try and sort of see what's happening with the sera and positivity of these COVID positive patients.
I mean, I'm just going to say that my guess is for something like this is that you're going to clinically order tests that are going to be able to test these things over time. And that's going to immediately, the biobank will get information in large numbers, but I don't know that it'll be as targeted and specific as what you need for this specific study.
Okay. Any other questions? A couple of folks unmuted there. Just, Chazam, would the biobank be interested in partnering with clinicians for prospective collection of CSF during elective spinal anesthesia for COVID screen patients in obstetrics?
I think the answer is undoubtedly yes. CSF just in general for me is something that I hear from investigators all the time. We never have in biobanks that would be very valuable.
Yeah.
It's a great suggestion.
Was there any other questions? I thought I heard somebody.
Yeah, actually, I have a question. This is Emily. Thanks, Wendy, Christoph. This is really such an exciting resource, and I know it's a lot of work that's been put into this. I was wondering, will viral titers be measured at all in any of the COVID patient samples?
So maybe I can answer this. So we're piloting on measuring the viral load from the nasopharyngeal swabs. We are also recovering viral genomes from the nasopharyngeal swabs for sequencing. So the plan is to generate this at scale and match it with the host genetic data. I don't know the precise results of the pilot yet. This is kind of still in a pilot phase. In terms of measuring the viral load in blood samples, we are also running a pilot where we extracted RNA from these samples, and we're measuring, the viral load. But I don't have the results yet. So we're actively thinking about this. We are not entirely sure that we'll be able to recover virus from the blood in sufficient amount for sequencing. So that's one of the issues. Nasopharyngeal swabs are probably preferable also because of the sampling, of the timing of sampling. So these are typically done in the setting of acute infection. So the chances of detecting the virus and capturing the viral genome are higher.
Great. Okay, I think that's all of our questions. Our next couple of offerings have been up on the screen for a while, so I don't need to read through those again, but please join us tomorrow and next Tuesday as we continue in our Cancer Center seminar series. It remains for me to thank our two speakers very much for joining us today and for their presentation, and to thank all of you for joining us and for a lively Q&A. I appreciate your time. Thanks everyone, and take care.
Thanks for having us.
Thanks.
Bye-bye.